it's already in the blood, we can excrete it in the urine or break it down in the liver or bind proteins to it and stuff so it can't bind to its receptor. Or, once it's already in, we can deregulate, downregulate the receptors so we don't have very many receptors. But if we already have the cyclic AMP in the cell, then we have to stop that. And so phosphodiesterase is an enzyme that does the opposite of the adenylyl cyclase. It breaks down the cyclic AMP. So phosphodiesterase is going to break some of the bonds between the cyclic AMP, the ester bonds, and it's going to decrease the amount of cyclic AMP. So there's a bunch of different steps that the cell can use to stop this activity if it doesn't want to keep these enzymes. Now what's it going to do? Let's say we already have active enzymes in the cell. Protein kinases have been activated and we phosphorylate enzymes and they're working. How do we get rid of those enzymes? Back to Bio 430. Pardon? What's the organelle in a cell that breaks down proteins?
Lysosomes. Phagocytosome.
Okay, so we can, the phosphodiesterase will get rid of cyclic AMP, but if these enzymes are already active, it's not going to take care of those, so those can be internalized by the lysosome. Okay? All right. So our second mechanism, so this is G proteins and cyclic AMP. Probably one of the most common, at least 50% or so are going to be using this particular mechanism. The second mechanism is a little bit more direct. All right, not quite so many steps. We still have the receptor binding to the hormone, the receptor binding, the activation of the G protein alpha subunit, but instead of binding to adenylyl cyclase or another enzyme, that alpha subunit directly opens the calcium channel. And you're used to, from muscle physiology, how calcium released from the sarcoplasmic reticulum binds to troponin in a muscle cell and causes that to come off of the myosin, the actin complex, and allow myosin and actin to bind together. All right? So this is the plasma membrane calcium channel, shown here. It's opening, and calcium comes in. Here it's binding to calmodulin, which is similar to troponin, and now this is our second messenger. So calcium, either by itself or as bound to calmodulin, can be a signal to smooth muscle, to other activities in the cell. This was not quite as complicated. The most complicated is the last one. It actually combines these two together. All right, so if you think of these, if you understand these two steps, the cyclic AMP and the protein kinase activation, and then the calcium ions, the last one that we're going to look at pulls those two together. That's why it's complicated, because it has these two components. So I may go over 10:15 a little bit, but I'll try to go a little slowly, and then we'll come back and review these three things first thing on Thursday. All right? But don't wait till Thursday to study. See if you can read up about it. So, and this isn't that complicated, it's just the words are foreign. Okay, the words are long and sound kind of weird. But what they have in common is protein kinase and calcium. So they both involve protein kinase and calcium. So, common step, receptor and G protein. All right? Our hormone comes along. And binds to the receptor. The G protein is released and the alpha subunit is activated. I'm going to leave the alpha protein in the plasma membrane, but remember that technically it doesn't have to be there. And it's going to use GTP to activated, but now it's going to bind to a plasma membrane enzyme. I don't want that quite that big because I need another enzyme, another molecule in there. It's going to bind to an enzyme known as phospholipase C and be activated by the energy of the G protein. So instead of adenylyl cyclase, our membrane enzyme is phospholipase C. So that's the first difference here. Okay? The alpha subunit is activated in the same fashion, but now instead of adenylyl cyclase, phospholipase C is involved. Phospholipase C now breaks down another membrane-bound molecule, which in some diagrams looks like this one. It looks like it's not in the membrane, but it is. And remember, this is purely random, this moving around and interacting, because this is a lipid bilayer. And so the active phospholipase C just randomly bumps into the next molecule, which I'm going to abbreviate as PIP2. This is the longest of our words, okay? So phosphoinositol phosphate, PIP2. And by the way, when you're filling out the homework, the lab report, or if you're writing out short answers for the lab exam or the lecture exam, you need to write out the whole word for full credit. Okay? PIP2 instead of phosphodinositol phosphate is just half credit. I want you to know what these words stand for. Now, this is the enzyme, this is the substrate. And PIP2 is broken into two molecules. One molecule is in the cytoplasm. The other molecule here looks like it's in the cytoplasm, but it really still remains membrane bound. Okay? And I'm running out of board space, so I'm going to, let's see. And everybody, since now I need more board space on that end, so let me just redraw this. Here's the alpha subunit bound to phospholipase C. Oops, one more. Probably should have put it up a little higher while I was at it. All right, so the effect of this enzyme is to split PIP2 into two molecules. One of them goes into the cytoplasm and is identified as inositol triphosphate, or IP3. The other one associated with the plasma membrane and is diethylglycerol. So PIP2 forms IP3 and diethylglycerol. Everybody with me so far? All right, so remember the two things that we were combining were the calcium ions and the protein kinases, right? So what inositol triphosphate does is, like the last process that we just talked about, it opens calcium channels inside the cell. So remember how muscle cells stored calcium ions in sarcoplasmic reticulum? All right, so inositol triphosphate is going to open intracellular calcium ion channels. So smooth endoplasmic reticulum channel sacs that have calcium ions in them. Those calcium channels will open. And calcium ions will be released. Okay? So that's the part of this pathway that is similar to this one, opening calcium channels, but they're intracellular calcium channels. And then calcium itself can be a second messenger. Now it's not very clear in this image here, but the calcium ions plus diacylglycerol can activate protein kinases, and these are called protein kinase C. So the IP3 does the calcium release that we saw in the second version of G proteins. The diacylglycerol can activate protein kinase C, similar to cyclic AMP did. So at the end result, we have the combination of those first two mechanisms. We have extra calcium ions in the cell, and we have protein kinases. Okay. Now those protein kinases may also open extracellular plasma membrane calcium channels, but this is the primary response. So now, anytime we have extra steps, it does two things, okay? It provides further amplification, so it increases the overall effect, the size of the response, and it provides extra control. It provides extra places where we can stop it, extra places where we can regulate it. Okay. Now, as part of the lab report that's due next week, I have a couple of these pathways written out with empty boxes for you to fill in. So just, again, as a heads up, try to follow through these pathways on your own first before you go to the homework and look it up and just fill in the boxes by looking up every answer. Okay? See if you can come up with it on your own. On Thursday, we're going to stop here. On Thursday, we're going to look at a shorter way in which the receptor itself will activate something similar to cyclic AMP without it. Its receptor is the enzyme. And then we'll look at where the receptor not only, in the previous case, the receptor is the enzyme that activates a cyclic GMP, and that activates a protein kinase. So the molecule that we're getting rid of here is the receptor activates guanylate cyclase without a G protein. So we're getting rid of that intermediate step of the G protein. The receptor has the effect of the alpha subunit. And then the second mechanism, what molecule have we gotten rid of here besides the G protein? And the membrane enzyme. What else have we gotten rid of? The receptor itself is acting as what enzyme? It's adding phosphate groups to other molecules. What do we call the molecule that did that? No?
Protein kinase.
Protein kinase. Okay, so in this situation, the receptor itself is the protein kinase. kinase. And we've gotten rid of the G protein. We've gotten rid of the adenylate cyclase or the phospholipase C. So this is a much more rapid response, but less controllable. Okay, we can't like lower other levels of other molecules and control it. So on Thursday, we'll come back and review what we just covered. All right, and look at these 2 steps. Talk a little bit about how we regulate levels, and then move into talking about the pituitary. So for those of you, anybody come in after I took roll, I gave a permission number to one person, the other individual, remember, I can give a permission number to. Let me check. I will put the rest of you on D2L.